Hey sweetest potatoes, so today we are going to study about homeotic selector genes in Drosophila. So, so we'll be covering a little bit of introduction where we'll know what homeo means, brief anatomy of Drosophila, and then move on into the topic that is homeotic genes and homeotic mutants, and finally summarize the topic. So coming to the introduction. So before going, so what do you mean by homeo? So homeo, it comes from a Greek word which means similar, and homeotic mutants are mutants in which one structure is replaced by another. For example, if your antenna of the Drosophila is fruit fly. So if the antenna is replaced by a leg, so then it is a homeotic mutant. And homeotic genes. So these are the genes which are very specific, and it is the identity of a particular segment. For example, a specific body segment will have a gene and which will have a phenotype. So that is homeotic gene. And then you have homeobox. So homeobox, these are conserved DNA sequence which are about 180 base pairs, and they're shared among many homeotic genes. And this sequence, that is this DNA sequence, that is the conserved DNA sequence, it encodes for about 60 amino acid, and that is referred to as the homeodomain. The homeodomain. And this homeodomain, it will go and recognize specific DNA sequence. And homeodomains are really important because they are important regions of gene transcription which are encoded by the homeotic genes. But you cannot say that all the genes containing the homeobox are homeotic genes. Okay, only some of them will become, some of the genes which are the homeotic which are present in the homeobox will become the homeotic genes. With this, we'll go forward. And now coming to Drosophila. So you know that Drosophila is one of the best understood organism. It has the best understood developmental system at gene level, and these Drosophila, so they have about 13,600 genes are present in the Drosophila. And these genes, they control the development of Drosophila, which are very similar to the development in case of vertebrates. Hence it is used as a model organism, and we are studying about it in detail. And if you take the the Drosophila larva, it is patterned with two distinct independent anterior as well as posterior as well and dorsoventral axis. And the larva, it has three anterior acron. So here you can see in the diagram, so this region, so it has three anterior acron, and then it has three thoracic region, that is T1, T2 and T3, and then it has 8 abdominal segments which starts from A1 up to A8, and each of these segments has its own unique character, and it is important, it gives an external cuticular structure as well as internal organization. And the sequence of events which takes place in Drosophila development is same as that of the C. elegans, which we have already studied. That is, there is fertilization followed by cleavage, gastrulation, axis formation, and finally body segmentation. And so yeah, with this we'll go forward. So coming to homeotic selector genes. So before that, what are these homeotic genes? So homeotic genes are those genes which regulate the development of the anatomical structure in an organism. And these homeotic genes, they encode transcriptional factors, and these proteins will affect the development by regulating the gene networks which are involved in body patterning. So mainly just you have to remember that these homeotic genes, they'll go and encode some of the transcriptional factors. Again, the transcriptional factors will give rise to proteins, and these proteins are important for the development. And the main function of these homeotic genes are involved in body patterning. So if that gene is mutated, then for example, a gene is mutated which your hands are supposed to be formed. Like if you're taking an analogy of yourself, and the gene which was supposed to give your hands get mutated, and you get your head in the place of your hand. So those type of mutations happen when the homeotic genes are mutated in case of these flies. And the mutations can cause the displacement of the body parts, which are very lethal to the organisms. For example, you see, each part of a body is organized in such a way that it's useful, right? So, for example, if you take this Drosophila, the Drosophila, the eye is missing, and instead of the eyes, you have the legs come in place of eye. So it becomes very lethal because it becomes kind of not function. The phenotypic expression is not functional for the organism to survive. So this is very important because it cause it it can cause even the death, and it's very lethal for the organism. And coming to this, the homeotic genes, specifically in Drosophila melanogaster, it was discovered by Edwin. It was discovered by Edward Lovis, and in Drosophila there are two regions. Okay, there are two regions on chromosome. There are two regions on chromosome 3, which contain the homeotic genes. So the first region is called as the Antennapeaia complex, and then you have the second region, which is the Bithorax complex. And there is also another region called as the homeotic complex. Homeotic complex. So the chromosome region, which contain both this Antennapeaia complex and the Bithorax complex, is referred to as the homeotic complex, or the home C. So we'll be looking into each of these complexes in detail. And also here, I've also there's also a diagram here where you can see. So in this diagram, you can see there is one complex called as Antennapeaia complex, which includes all these genes, and also the location of the genes are present. And then you have the Bithorax complex, and the genes which have both of this is referred to as the home C. So now coming to the first one, that is the Antennapeaia complex. So the Antennapeaia complex, it has the following genes, that is labial, Antennapeaia, sex chrome reduced, deformed, as well as proboscopedia. So now coming, so these genes, so if it the labial gene as well as the deformed gene. So you can see here the labial as well as the deformed gene, they specify the head segments. Then you have the sex comb reduced, as well as the Antennapeaia gene here, the sex chrome reduced, which is abbreviated as SCR. So these two are involved in the thorax region. So they are very important for the thorax identity, which you can see in the diagram. And then you have another gene which is referred to as the proboscopedia. So the function of this proboscopedia, you cannot see it here because it acts only in adults and not in the larval stage. And if this gene is absent, then you can see the labial palps of the mouth is transformed into the legs. So you can see legs in place. Where the mouth part should be present, you can see legs instead of that. So this is all about the Antennapeaia complex. And now we'll go to the next one. That is the Bithorax complex. So the Bithorax complex is made up of the following genes: the Ultrabithorax, abdominal A, abdominal B, and home C. So the Ultrabithorax, here again you can look at the diagram. So we are talking about this complex, that is the Bithorax complex. So the UBX, that is the Ultrabithorax, it is required for the identity of the, here you can see the blue color, the green color here. So it is required for the identity of the third thoracic segment. And you have abdominal A and abdominal B. So these both are responsible for the abdominal identities. Here you can see the blue colored region. So it is required for the uh, here they're required for the abdominal identities, whereas the UBX is required for the thoracic third segment identity. And as I've already told you, this complex, that is the home C, it is the region of the chromosome which contains both the antennapeaia as well as the bithorax. And you know now that the homeotic selector genes is responsible for the specification of the fly's body parts, and if there is any mutations in any of these genes, it will lead to bizarre phenotype. So now we'll look at all the mutants. So in the year 1894, a person named as William Bateson, he called these organisms as homeotic mutants. So now, if you take a normal fly, here you can use the diagram for your reference. So if you take a normal fly, so it has three thoracic segments, that is T1, T2 and T3, right? So all the thoracic segments, here you can see the diagram again, the three thoracic segments will give rise to the three pairs of legs. So now to go in detail, so the first thoracic segment, that is the T1. So the first thoracic segment, it only gives the leg. You can see here, it only gives the leg. Whereas the second thoracic segment, it gives a pair of legs and also it gives a pair of wings. It gives a pair of legs, and that is T2, it gives a pair of legs and pair of wings. Whereas you have the T3. So T3 is responsible for giving a pair of legs and also a pair of halter. So as you have already done some experiments on the Drosophila. So halters are when you do the dissection and all that, you can see them in imaginal disc. So halter are the balances. So these halters are seen in the thoracic segment 3. So I want you guys to remember this, because then we'll know what happens in case of mutation. So the first one is the Ultrabithorax gene mutant. So in Ultrabithorax gene mutant, when this Ultrabithorax gene is deleted, this Ultrabithorax is very important for the third thoracic segment. So it is characterized by the third thoracic segment, and in normally what does the fly form? In the third thoracic segment is important for a pair of legs and also a pair of halters, right? So now this gene which is responsible is mutated. So when this gene is mutated, then it is seen that the third thoracic segment T3 is transformed into T2, that is the second thoracic segment. So what happens? So what did we see previously? That is the second thoracic segment will produce um, a pair of legs, and it produces a pair of legs and a pair of wings, right? So now what do you see here? The third thoracic segment is converted into the second thoracic segment. So this results in the fly with 4 wings. So the halter is gone because it is mutated. So instead you'll see, instead of 2 wings, you can see 4 wings. And this is like very embarrassing for the fly, because uh, the classic 2-winged fly, and they're also referred to as diphtherians because these insects have only 2 um, winged insects. And then when this situation happens, when this Ultrabithorax gene is mutated and the third thoracic segment is converted into the second thoracic segments, and the halter is absent. Instead of the two wings, as you can see here, it will have two pairs of wings. You can see four wings. So that is about the ultrabithoracic gene. Now coming to the next one, which is the Antennapeaia mutant. So in Antennapeaia mutant, so this is very important for the second thoracic. It specifies the second thoracic segment of the fly. Again, going back, so what is the second thoracic segment? It produces a pair of legs and a pair of wings. So when this mutation happens, uh, the Antennapeaia will get expressed in the head as well as in the thorax. So the actual function was only in the thorax, but now it got mutated. Now we can see the expression of Antennapeaia also in the head. So in so the you can see here in this diagram, instead of the antenna, the legs will start growing in the head sockets. So that is the mutation. So one of the reason to explain this is that, so you have this Antennapeaia protein. So the Antennapeaia protein, it will go and bind to uh, two enhancer genes, which is referred to as the homothorax, as well as there is another gene which is called as the eyeless gene. So this Antennapeaia protein, which codes for the transcriptional factors, and these are very important for the formation of antenna as well as the, so you have the homothorax gene as well as the eyeless gene. So these are important for antenna as well as eye formation respectively. So when this Antennapeaia is mutated, so this will go and suppress the genes which will trigger the eye and the antenna development. So because of that, because of suppression of these genes, you can see instead of the antenna, the leg is produced in the eye socket. So this is in case of dominant mutant of Antennapeaia. So if you have a recessive mutant, of Antennapeaia, then the gene fails to express in the second thoracic segment, and you can see antenna in the leg position. It's like the reverse. So here you can see antenna in the eye position, instead uh, the legs in the eye position. But if it is a recessive mutant, then you can see the reverse of this. That is, you can see the second thoracic segment. The antenna will be seen in case in the region of where your legs are present. So I hope you guys understood, and here are some of the diagrams. So these are the mutations of Ultrabithorax, which is produced in a fly, and you can also see the mutations of Antennapeaia, where you can see the legs in case of the antenna, and in case of recessive mutant, the reverse happens, where you can see antenna in the region of legs. And this is an example for mutation of Drosophila, which is seen in case of adult flies, where the mouth parts are converted into legs, and this is a labial mutant. So with this, I would like to summarize by saying that the homeotic selector genes are found in two complexes. That is, you have the Antennapeaia as well as the Bithorax complex on chromosome 3 of the Drosophila. Together, these regions are called the home C, or the homeotic gene complex, and the genes are arranged in the same order as the transcriptional expression, and the home C specify individual segments and mutations are capable of transforming one segment into another. So if there is any mutation, the function or the phenotype also changes, where it is converted into another segment. So I hope you guys understood, and if you like this video, please click a like and consider subscribing for more awesome stuff, and thank you so much for listening.